Thanks for joining me for this bonus episode of the Maine Science Podcast. I'm Kate Dickerson. We have something a little different today, a conversation about the discovery of a new species of tardigrade found in the Bangor City Forest. Tardigrades are microscopic animals and are really remarkable creatures. We'll have more about them in the episode. In this episode, I talked to Dr. Emma Perry and high school student Noelle Killarney, the team that discovered this species. We also talk about Emma's background, since it played a big part in how she got into studying tardigrades. Emma and Noelle would like listeners' help in naming the new species. I've included a link to that survey, which closes at 5 p.m. on October 30th, 2020, in the show notes. One more thing to note, we actually went to Bangor City Forest to have this conversation in a physically distanced, appropriate way. So you will hear a little bit of wind and background noise, including a helicopter during the audio, but it shouldn't be enough to distract from it. And finally, a big thank you to Emma and Noelle for their patience in getting this done. Due to some technical difficulties on my part, it took a little bit longer than I had anticipated, but I am deeply grateful for their time. I hope you enjoy the episode, and don't forget to vote for the tardigrade name. We are walking through Bangor City Forest. Emma and you talked about what a great refuge it is in general, but during pandemic times in particular.
Yes.
And the main reason we're here is to talk about tardigrade.
Yes.
Which I think are among the coolest creatures ever that I've heard of. I only first heard of them a couple of years ago. So maybe you can give just a quick background on what tardigrades are, and then we'll talk about why we're here to talk about them.
Okay, so tardigrades are also known as water bears. They are very, very small. They are about, the very, very biggest are maybe 1 millimeter long, but more typically they're a little under 0.5 millimeter long. And they live on the water layer around moss, lichen, in leaf litter, in the soil, on the sand and the beach, in between barnacles, pretty much everywhere. And if you really want to think about it, because it's been so dry here in Maine, and we know, lichen and stuff, it gets dusty, you've probably swallowed a few tardigrades blowing in the wind this summer.
We could say that about almost everything in the air at this point, right? Of what we've swallowed. So you said they're everywhere. Is there any evidence about how high up they can, like if someone was on Everest, would they see a tardigrade?
No one sampled.
Really? I wonder if there's any way they could retroactively check. Because are there samples from Everest?
No.
Of anything?
I don't know that biologists have gone up there. There is an association now of explorers who reach out to scientists to say, hey, we're going there. There are samples on glaciers.
There are.
In the criconite holes. So that is on a glacier. It's white, it reflects the light, but the world is a bit of a dirty place. So there's always a speck of dust or something, and the dust is black. So then it gets hot and it melts, and that attracts more dust. And so from a distance in the movies, it looks beautiful, shiny and white, but in reality, there are places, these little sort of craters that have collected dust, and down in there's a whole little community of small animals of meiofauna, including tardigrade.
I just went right to a Pixar film, right? Like, how cool would that be if that was a Pixar film?
And there's even special species that are only found in those criconite.
So is that because of the dust that they get there? And how did they get there?
That's a question. They've probably been blown. There's some of their distribution seems to reflect the air patterns. But they've also been found on the feathers of birds. And it's a possibility, if you think about where we are, they could be picked up by the underfur of other animals as well.
And that's how they got on the glaciers?
And the glaciers, possibly from birds, or also just blown in the wind.
All right, so tardigrades everywhere.
Yes.
Microscopic.
Yes.
We've eaten them in all likelihood this summer.
Yeah.
All right, that's fine.
Probably haven't tasted them, they're too small. Nah.
So we're in City Forest. Why are we here to talk about tardigrades?
We are here because we discovered a new species, and we would like your help in naming the species. We've got it down to 4 names, and we just need your help to pick the final one.
Okay, so before we go into that anymore, how cool is it that you both discovered a brand new species? Like, I'm going to guess you didn't go into this thinking that.
No. I went into it thinking that I was just collecting samples to see what different species were found here and if there's like a variety.
So it was just purely like, let's just see what's there.
Yeah.
Then we'll identify them and compare them with other places where we know it happens, right? Okay. I mean, I just think that that's like, if you love science at all, or have any, I don't know, fangirlish is the way I think about it, the fact that you get to say, I found a new species is pretty awesome. So what makes this species, why is it so interesting in particular? I have a vague memory that you had said it's an unusual tardigrade species.
It's a pure freshwater one, meaning that it doesn't, as far as we know, survive the drying out because it's used to living in streams, so it's a stable environment.
So maybe we should back up. This is one of the reasons why tardigrades are so cool and they can be found everywhere. Yes. They need water to live, right?
Yes.
That's the water bear part. But they can live without water how?
So tardigrades are active in a water layer, even in sort of moss and lichen. They don't live in the moss itself. They may eat it, but after it rains, moss will trap the water, and the tardigrades will crawl around in that water layer doing their thing. As the sun comes out and the place warms up and the water evaporates, those tardigrades survive until the next rainfall by going into a resistant ton. And essentially they allow themselves to dry out. They suck in all their legs. They pull in their sort of hind legs up, and they pull in their head until they sort of resemble an old oak barrel in shape. And in that form, they're pretty much inactive. Think of being in stasis like in the old Star Trek episodes. And to our knowledge, very, very minimal metabolism occurs. So if you were measuring how long a tardigrade lives, pretty much you don't count the time they're in the ton. So it doesn't affect how long they're active. And then the water comes back and they rehydrate and their legs pop out and their head pops out and off they crawl again, doing their business.
So have you seen tardigrades do, this is a total separate question, because if you have a sample with water and you slowly take the water out, will you see the tardigrades do this?
Yes.
Seriously? Have you seen it?
No, I haven't.
Now you want to though, right? Because that sounds really cool.
I'd seen bits of it, but I was teaching a course for Eagle Hill this summer on tardigrades, and a lady was taking the course, and she saw it right under her microscope. And she sent me pictures, and she'd taken, like, she had put times. So she's like, at this time it did this, at this time it did this. And then she added the water back, and it came right back. It was very awesome.
That is really cool.
And it can happen relatively quickly.
And if I remember this right, one of the coolest parts about tardigrades is, besides this ability, they can survive for a really long time.
Yes.
In all sorts of environments, right?
Yes.
Frozen, water. Well, I guess the key part is at some point they need water to come back.
Yes. If they're in that resistant ton, they have specimens that were over 20 years old. They were in the freezer. They pulled them out. They added water. They came back to life. Also in the ton, they sent some up into space and exposed them to solar radiation and the absolute vacuum of space, and they came back to life.
Seriously, the vacuum part?
Mm-hmm. Now, not the ones that are out active and crawling. Those were done, but the ones in the resistant ton. And they are variable. I won't say every species, every tardigrade survives equally. But the limnoterrestrial ones tend to.
And those are the freshwater ones?
Those are the ones that live in the moss and the lichen. The ones that live in the intertidal zone, who are in the ocean for part of the day and dry for part of the day, they do it. But the ones on the bottom of the seafloor, and the ones who are constantly at the bottom of lakes and rivers don't.
Because they don't need to.
Exactly.
Okay. And that's what you two found, is one of those.
Yes, because Noelle and I were chatting about possible projects, and there's only been a couple of papers in Maine, and I've added a lot of species, just what is here, since I've been working, but nobody's really looked at freshwater streams. So I just suggested to Noelle, it was one of the many ideas I threw out, and Noelle liked it.
And Noelle, this is why you thought you were just going to find ones that were already there.
Yeah.
Right. Do you have any idea how many species of tardigrades there are? Does anybody?
There are approximately 1,300 right now.
Based on what we know, or is that a projection?
Nope. Those are described species. We believe there's probably at least 2,000. So there's a lot that are undescribed.
So this tardigrade that you're gonna show us where you found it, how would you describe how it looks? Water bear for me brings a very prominent, like when you watch videos of bears just kind of walking along, like that's where I go to with this tardigrade. Would you describe it like that, or is it a different animal that we can compare it to?
No, kind of like that, but with like double the sets of legs. Because it's got 8 legs.
And do they all have 8 legs?
Yeah. That's one of the unique features of the phylum. That's how we know.
But they're not in the same category as spiders, or are they?
No, they don't have a rigid cuticle like the arthropods. So they do molt, but not that. Molt more like a snake, not like the whole insect thing.
Okay. So Noelle, why were you looking for a project with Emma?
Because I wanted to do the science fair, and so my dad mentioned something about how she studies tardigrades, and she'd probably be willing to help me with the project.
Did you know what tardigrades were before this?
Yeah.
How come?
Well, not exactly like tardigrade itself, but I've heard them referred to as, again, water bears, and I've seen pictures of them before.
Was this the first time you had the chance to actually search for them yourself and look at them under microscopes and that whole thing?
Yeah.
Okay, so there's this brand new species that you two found. Noelle, you sampled, and Emma, you cataloged it and figured it out.
Yeah.
So how long did it take from sample to, wow, we have a new sample or a new species? Because it's not instant, right? You're not...
Yeah, because I think I took this sample last November, and it wasn't until kind of recently that she was able to identify it.
We thought it was a new species pretty much right away, but you have to confirm with your friends. And then they were like, well, yeah, we think it looks like a new species, but I needed to do the formal description, the measurements, etc., just to confirm and get some DNA evidence as well.
How do you get DNA out of such a little tiny creature?
You put it in a little vial with some proteinase to digest away the protein, and I think the total volume in the end is 50 microliters, and then you PCR it.
PCR is?
Polymerase chain reaction, sort of like a photocopier for DNA.
And that's the test that everyone says is the gold standard for the COVID test, the PCR tests, right? Because they're finding the DNA.
Yeah. Yeah, not an echo of it, but the real thing, yeah. And so you choose the primer. It doesn't amplify the whole genome, just the piece you're looking for.
So since we have a little bit of time, anybody who's listening to this is going to realize that you're not a Maine native, based entirely on your accent. So if you could give us a little bit of a background of who you are and how you got into science and how you got to Maine.
Okay.
And if that's related at all, that would be cool to know as well.
So I was born in the southwest of England, the land of Jane Austen and Daphne du Maurier, etc. And I always wanted to become a marine biologist. So I went to the University of Exeter, down in the southwest of England. And I did biology with sort of a marine focus. And then I applied to do an internship at marine labs all around the world. I mean, I sent some off to Australia by the reef. I sent some to America, etc. And a few people replied, including Dr. John Lawrence from the University of South Florida in Tampa. And he said, no, I don't have funding for a summer internship. How about coming and doing a PhD? And I said, well, why not?
That is not the normal route that most people take for a PhD, to jump up a level from an internship to...
Well, yes and no, because in England, typically, at least then, you typically would do your undergraduate degree and go straight into a PhD. In England, you only did a master's if your undergraduate wasn't good enough. Is an intermediate step.
I see.
So I didn't know that I was doing anything abnormal.
I think the unusual part is the mental switch you went from, summer internship, sure, PhD program, because the length of time of those two are pretty different.
Well, there was a difference. I mean, the summer internship was between those degrees are 3 years in England, and I'd applied for the summer of after my second year, so I would have had a year left. And that was good because I had to do all the applications by snail mail back then. So, I mean, it took time. It took time to get the visas. I had to arrange. I mean, I spent a day in the doctor's office, and because I'd moved around a lot as a kid, she looked at a list of all the shots that I needed for the U.S. immigration, and just said, I don't have your record here. Give me both arms. And she shot me with all of them. It's all right. I was young and silly. I don't remember any pain.
Plus, you got to come and do marine science.
Yeah, so I went to the USF and I studied how sand dollars made their skeletons. The mineral they're made of, if you were to grow the crystal in a beaker, would grow just like a brick. But the echinoderms put proteins in the mix, and that sort of morphs the crystal. And they're famous, echinoderm skeletons are famous for acting more like glass, so they break with a conchoidal fracture.
I'm always fascinated by people who take inspiration from nature and apply it, right? And that strikes me as another really good example of ways that you could learn what critters are doing, and they don't, as part of their evolutionary bit, and we can learn from it.
Oh yeah, they're also famous because, they really don't use their muscles for too much. They walk and do everything with their tube feet, which is more of a hydrostatic system.
Oh.
After that, Florida was too hot after grad school, so I deliberately looked for a job back up in New England, and I got to come to a small liberal arts college here in Maine, and I've been here ever since.
And you've been doing marine science, and
Yes.
Why tardigrades? How did you, tardigrades and sand dollars you can see. Tardigrades not so much without some help.
That's true, but I was teaching invertebrate zoology as part of the marine biology major. And invertebrate zoology happened to be a spring course in spring here in Maine, 6 foot of snow on the ground, and all very, very cold. And so the college I was at, students, we tended to be very active. People are used to going outside, and so you get to about the end of February, and students tended to get a little stir-crazy because they couldn't go outside right now. And so they weren't paying attention in that particular class, and we had gotten through the invertebrate textbook to tardigrades. So it was really pure serendipity that they were in that mood. I looked down. It said in the textbook that if we went and collected, they would be alive. So I said, okay, go put your coats on. And we walked outside the classroom into the woods, which were only about 100 yards away, collected some moss and lichen, added water, and then looked at them the next day, and there they were. So that was really exciting. Yeah, and then I did a little research, and so you go and look and you say, what is the first tardigrade we've seen in the States? Who found the first one? And it turned out to be that the first tardigrade recorded in the States was collected by a priest from New Gloucester, which is just above Portland, Maine. And so when I saw the first tardigrade had been collected in Maine, it's sort of a sign.
You have no choice.
Keep studying tardigrades. And it's such a fun group to work with because they've been kind of ignored because they're small.
Sure, and you can't see them with your eyes, and we're only just figuring that out. And so that leads us to here. Not only is it a fun group to work with because we don't know much, but you get to find new ones, right? So it's very dry right now.
I know.
And these are the ones where they need water.
Yes.
We would just see eggs.
In the sediment.
And what do the eggs look like?
If you are a person of a certain age, they would remind you maybe of Sputnik. So if you are not of a certain age, think of a very large beach ball and then stick cones all over it.
Have you seen both the tardigrade and their eggs under the microscope?
I've seen tardigrades. It's just that I probably have seen eggs, except I just don't have enough skill to be able to recognize.
Yeah, that was my whole experience with every microscope I've ever looked at. It was just brutal for me. I just knew that biology was not going to be my thing for that reason alone. So have you seen your tardigrade?
Yes, because I was the one that had to put it on a slide and make sure it was...
The very first tardigrade Noelle found was the new species.
I wouldn't say it was the first one that I found, but it was in the first sample I collected. Mm-hmm.
That's really cool. Well, you can only go downhill from here, so...
Yeah.
Or you can be the one who discovers many, many new tardigrades from here on out.
Yeah, so we were always going to work through it, and then we had the pandemic. Yeah. And I was working with a colleague up in Orono learning how to sequence, and I got kicked out of the lab along with everybody else, right? We all got kicked out of everywhere.
Yeah. Yep.
And of course, we all got told to stay home, but we were allowed to go out for walks and hikes. And so as I knew the site was here, it was an ideal time I could come and collect. And I could make slides and do all the morphometrics. So you have to measure everything to describe.
We had a really wet spring too. Yes. Did that help?
Yes, it was lovely. Yeah. And this is, it has a name of a river, so it is generally thought to be mostly wet. But we're in drought right now.
Yeah, we're in a serious drought. This usually is damp year-round, is what I remember it. And you're right, in the spring it's
very wet.
Really robustly wet.
And so, it seems odd saying that this is where a very wet species lives.
So do you too, this is pure conjecture, the odds are good there's other new ones out here, right? Here specifically, right? Probably. Just based on the type you said it was, and that there's so many hundreds of species, right?
We have no records of freshwater tardigrades from Maine, which means if there are species out there, but they're not recorded in Maine for the pure freshwater ones. So yeah, just a hike out there into the wetland where the beavers have dammed could undoubtedly give us a whole bunch of new records anyway.
Okay, so the contest, which is the other reason we're all getting together, is we have 4 names to choose from for this new species, because if you find a new species, you get to name it.
Yes.
Right? So we are going to put it out there for Mainers to name this new species, and then we'll announce the winner. You did the research, you did the sample last November. Mm-hmm. I think the turnaround time's pretty fast for a species. I don't know how this works.
Well, I've still got to write the manuscript, but I've already consulted with my colleague. And then it'll have to go through the peer review process. But yeah, it should go pretty fast as long as I sit down and write.
That is the hardest part. That's one of the reasons that got me out of a lab was having to do that kind of writing.
But it's pretty straightforward. So yeah, they're not big papers, but they're really important. You can't really understand how the world works if you don't know what animal you're working with.
So that's the other question I'll make you answer. Why should we care about tardigrades? We can't see them without help. They're everywhere and we don't even know it. What is the value beyond the super cool part of we are really surrounded by all these critters all the time and we don't even realize it.
Well, that, and I really think we should not try and make everything is going to save human life. I mean, not everything is going to be that important. I think we should acknowledge that it's good to be curious and that there's so much of the world we just don't understand.
And I agree with you that not everything has to have this huge, deep impact on human life. However, I also think there's a lot of things happening all around us all the time that we don't understand how it works, and we have no idea what the impact is.
Let me put it this way, all right? Everyone learns the triangle of energy with the trophic levels in some sort of biology. So you get the grass or whatever grows, and then the herbivores eat it, but they can't use all the energy, it's inefficient, and so on and so forth. That's the basic thing that everyone learns, but that completely fails to incorporate all sorts of things like parasites, like recycling. So when you die, you don't just rot back to inorganic carbon and have to be fixed again by a plant. You might be eaten by a bacterium that gets eaten by a worm, that gets eaten by a tardigrade, that gets eaten by something else. Or the trees, and in the trees it could even be the canopies, stuff never gets to the ground. All these little myofauna will recycle, or they'll compete with the bacteria by eating them to stop diseases happening. Or even the little water bear walking around in the leaf litter in the soil might even spread disease. We don't know. And not just tardigrades, other little myofauna. We don't look at the myofauna as much because they're small and people don't like looking down microscopes.
And it's a skill, right? I mean, you have to work at it and it's hard to get and you need to have a good teacher. I mean, all of those things are really important. So, well, I like to think that this is our small little step into making people excited about tardigrades in general.
And they're cute.
They are terrible cute. I really like them. Microscopic life in particular. And the fact that we get to be we have been home to a brand new species. I think it's pretty awesome. Thank you for doing this again. I appreciate it. The Maine Science Podcast has received support from the Maine Technology Institute and is recorded at Discovery Studios in Bangor, Maine at the Maine Discovery Museum. The Maine Science Podcast is produced and edited by me, Kate Dickerson. I received production support from Maranda Bouchard, and the Discover Maine theme was composed and performed by Nick Parker.